Welcome back, everyone. Our next session is on interactive taxonomy visualization. Please welcome Mr. Karthikeyan, a project associate at Brick National Institute of Immunology with expertise in bioinformatics and web development. His work focuses on building tools and applications for metagenomic analysis and interpreting complex biological data. He will now introduce us to interactive taxonomy visualization tools and approaches for understanding biodiversity data, and as well as a hands-on DIY session where you will actively engage with tools and techniques for biodiversity data. Over to you.
Okay. Hi everyone. Thank you for the opportunity to present my work. Huh? Is it fine? Too fine? This works? Okay, all right. Okay, again from the top. Okay. Hello. Yeah. Okay. Hi again, everyone. So, quick overview of the session. We'll quickly look at what metagenomics is, what type of data you get from metagenomics, why it's important, and then how we'll visualize it, right? So, starting off, role of microbes. Everyone knows microbes are everywhere. But then what a lot of us don't realize is everywhere they are present, they have very important functionality in the background. Like for example, in ecological succession, right from when it goes from bare rocks to lichen, an intermediate step is always microbes. They help the formation of a biofilm, which helps break down the rock, and then that leads to further succession. But their work does not stop there. After that, in between phases when we are switching from, short shrubs to higher organisms, they play an important role in the decomposition to help maintain. Sorry. Yeah. They help in decomposition and, they help maintain the nutrients in the soil. And further down, when the environment matures, they help stabilize it, keep everything stable. They do a brilliant job in the background. Similarly, in the human gut, they play an important role when it comes to digesting plant dietary fibers. We are normally not able to digest plant fibers. Specific bacteria convert them to short chain fatty acids, which are then converted to energy. Otherwise, we will lose a significant amount of energy gain, which we get from plant fibers. In fact, our human body considers these bacteria microbiome so important, we have a snapshot of them in our previously considered vestigial organ, the appendix. But do we know enough about them? I don't think so. The challenge of studying these microbes. So what do you think of when you think of a microbiome, like a complex environment, right? This is what I think of it as. Of course, this is not a microbiome. This is a picture of a neuron, which is supposed to be a nebula. But this is how I like to imagine it, right? It's this complex mess of different types of organisms, bacteria, fungi, viruses, higher plants, their roots, maybe even animals, the host you find them in. It could be all of that. But then this is how I like to interpret them. So based on this, what are the challenges? So the first thing is trying to emulate something so complex in a petri dish, right? So one of the main things is the nutrients. So let's say the brighter regions are nutrient dense regions in the environment, and then the dark regions are nutrient sparse regions. That does not mean bacteria don't exist in the dark regions. They exist in both places. It's just the different types of bacteria, different type of fungi that exist that draws this gradient, and we can't emulate that. And then their deeply symbiotic nature. Because of how convergent evolution works, let's say we are looking for bacteria which can fix nitrogen. That's really useful for agriculture and maintaining soil quality. If we try the classical method of isolating only bacteria which fix nitrogen, we'll pull out maybe thousands, maybe 10,000s of species because all of them do the exact same. And it won't just be bacteria, it'll be both bacteria, fungi, virus, anything which needs to fix bacteria to survive. And the last thing is the sheer volume of species. Since we'll pull out so many, we can't efficiently culture or isolate so many species every single time, which leads us to the solution is to sequence them all. That is metagenomics. So in one sentence, metagenomics is the study of all genetic material in an environment without trying to isolate or culture the organisms. Back in 1998, a group of scientists were the first people who, instead of debating how to culture, how to isolate these bacteria, they said, let's just sequence them all. Let's treat them all as a single genome, a metagenome. That's where the entire field came into existence. So what started in 1998 over the past 30 years has had very important milestones. The first one being, of course, the conceptualization back in 1998. Then we had the Sargosa Sea shotgun metagenomics experiment, which was the first of its kind, where they went out to Sargosa Sea, collected almost 100 samples, and sequenced all of them. And this is in the pre-NGS era, which is next generation sequencing, which meant sequencing took a lot of time. So that was a huge milestone. And around 2006 to 2007 is when NGS was invented. It became more mainstream, and fields both bioinformatics and metagenomics had a big impact. So which brings us to 2010, when we first categorized the gene catalog for the human gut. Here we didn't figure out what species existed. What we figured out was all the functional pathways that take place in the human gut, which aren't human related, which the microbiota does. And then in 2017, we standardized the requirements for MAG. MAG stands for metagenome assembled genome, right? So what started out as what is there? Can we see anything? We came up to the point where we could extract full genomes without culturing anything, right? Just from the amount of data we're able to generate, we were able to extract full genomes that we had to standardize what classifies as a good quality metagenome assembled genome. And then coming to 2024 and the present, we have evolved from what is there, what function it does, to can we use it in therapeutics. There are studies which show that we can uh, we can use the composition of our gut microbiome to see if we are susceptible to uh, certain diseases like IBS, or some studies also suggest a cross-link between depression and your gut microbiome. So that has been the history of metagenomics so far. So how do we get started with metagenomics? First, you pick your sequencing approaches. So we have the short read sequencing approach. This was the first NGS method that was standardized, became mainstream. So what we do, we extract the DNA from the sample, then we chop it up into predictable sizes, usually 150 to 300 base pairs, and then we add our primers and then we sequence them. So through this method, we get sequences which have really high base accuracy, meaning the sequencer is confident that whatever sequencer says is what is present in the sample. It has a really high efficiency when it comes to cost. So the amount you pay versus the data you get out is really nice. But then what we miss out on because of the short length is its accuracy when you try to put it together. Because end of the day, you can't make sense of genome with just tiny pieces. You have to piece it back together, make a genome out of it, and try to understand what it means. So that's where its shortcomings are. So a more recent approach are long read assemblies. Here, instead of chopping the genome up, we just sequence it all together. And this, while easier to assemble, and also captures a lot of complex genomic patterns. Like we all know DNA is not consistent. It's not consistently variant. We have a lot of long spans of repeats where it's just 80, 80, 80 for 50 or 50 or 70 base pairs. And in case of short reads, we tend to lose that data. Long reads helps us capture that. But this comes at two costs. One, it's a tad bit more expensive than doing short reads. And the other is the low accuracy per read. Because we are sequencing a large chunk of DNA, there tends to be a lot more error if you compare a short read to it. So there is a third approach, not widespread in metagenomics, called the hybrid reads, where um, we do both types of sequencing on a sample, and then we use the long reads as our scaffold, and then try to fix any errors in it using our short reads, because our short reads are more reliable. So now that we've picked an approach, let's move on to data generation. We pick an environment of interest, we collect and preserve samples, collect the metadata, extract DNA from the samples, perform QC, make sure we are getting good quantity of DNA out from the sample, make sure it's concentrated enough. Then we prepare the library, which I'll discuss in the further slides. Depending on the methods you use, you would either use primers or adapters, and then we sequence. After we sequence it, the output are raw rates in the form of FASTQ. FASTQ is just FASTA files with quality metrics for each base predicted, that how confident the sequencer is that this base is actually the base it says it is. But as with any raw data, we are supposed to clean it up. We have to remove the primers and adapters from the sequence because those are non-biological. We remove low quality reads, basically reads with low accuracy. Low complexity reasons, what I discussed previously in long reads, the reasons which are captured in some analysis, we don't need them. So we try to remove them. They're only useful when you're trying to reconstruct a genome. And then chimera. In short reads, there is a tendency that two different sequences sort of merge together in your output. So we try to remove them. So uh, focusing on the steps 5 to 7, we have two different methods of sequencing. Either we can pick amplicon sequencing or shotgun sequencing or metagenomic sequencing. In case of amplicon sequencing, what we do is we try to focus on a tiny part of DNA, a tiny region which is highly conserved across organisms. Example, in bacteria and archaea, it's 16S rRNA, or in case of fungi, it's ITS. And then we amplify only this region using PCR, and we sequence that. And in case of shotgun sequencing, everything is sequenced. We're not trying. We usually you don't do PCR for shotgun sequencing, so this also preserves their relative abundance. You know, it's like taking a picture of the entire microbiome, although it's fairly missing. And we end up with reads from all type of organisms, fungi, archaea, bacteria, everything. Right. So now that we've picked the methods and then we have the FASTQ files, we move on to the taxonomic analysis bit. So what we do, our FASTQ files have reads. We don't assemble them. We use a reference database which has a bunch of known genomes. And then we try to align our reads with these genomes. And if a read aligns well with these genomes, we assign the reads the same genome. So here you can see we have the blue and orange genome. And if you can see, out of the 5 reads which aligned, only 4 and 3 were assigned. That's because in reality, not a lot of reads align with our reference database. And that is one of our shortcomings of taxonomy, is that we are limited by how much we know about the soil, depending on how big our reference database is. And the output of this are abundance table, which looks something like this. On the left side, you have the entire tree of classification. And then on the right side, you have read counts. And this is only the first maybe 7 or 8 lines of the table. These tables goes on for thousands of lines. And if you're lucky and the environment is well documented, it can go on for 10,000 lines. So that makes it pretty hard to interpret, right? So which is why we come to our visualization bit. What's the point of visualization? As as with anything, the main point is making it human readable. We don't like tables, we don't like a lot of text, we don't like numbers. But alongside making it human readable, in case of taxonomy, we are trying to capture these five data. Main is the high number of taxa and then multiple samples. So as with any study, it only makes sense when you compare it with something, right? So the ability for a visualization to make it easy for that comparison to happen is important, and relative abundance. Uh, because because of how taxonomy assignment works, not all of the taxa is assigned. So let's say we pick the soil environment, right? And we try to assign samples, assign taxonomy to it. About 30% of it will get taxa assigned, while the rest 60 to 70% remains unclassified. So we can't take absolute abundance. We rely on relative abundance. Relative abundance is just the total reads and then uh, under the reads of the taxa you're interested in. So it's a percent value. And then hierarchical relationships. Taxonomy inherently is very hierarchy based. So it is essential to capture that. And the last one is composition and structure. Well, what it means is both relative abundance and taxonomy should be captured together at the same time. So it makes more sense. It'll make more sense when I give you examples. So the criteria for good visualization is shows relative abundance, preserves the hierarchy, makes it easy to compare across samples, and remains interpretable at scale. So we'll start off with bar plots, the classic example for any type of visualization. Everyone's familiar with bar plots. So here we have a version of it called stacked bar plots. And in the example here, um, this is the composition of 17 odd soil samples at the rank of phylum. It goes from the highest composition to the lowest composition, and the red bar indicates unclassified. So pros, it makes cross-sample comparison very trivial, and we can easily find out what the dominant taxa is. And it's very easy for people to understand. Limitations, we completely lose out on taxonomy information. While we know a certain phylum is the most dominant, we lose information on what its subsequent children are or what its parent was. And we lose out on rare taxa because it gets lost in the numbers. So moving on, we have chronoplots. This right now is the standard for having an overview of a metagenomic sample. What it is an interactive sunburst plot, meaning it's a radial plot where the middle of the plot indicates the highest level, which in our case would be kingdom, and every subsequent lower taxa is placed radially outwards. And because it's circle, the composition sort of stays proportional, even though this might seem like the lower taxa is occupying a lot more space. What it's trying to depict is that they fall all under the same angle. Pros of it. It's extremely good at preserving taxonomy. If anything, the interactive part lets you drill down on a specific taxa. Let's say you're interested in a, a class over here. Yeah, let's say you're interested in this class, right? You can click on it, it would zoom out, and you would get a plot very similar to our current plot. Sorry. But yeah, so that makes it exceptionally good at interaction. But its limitations come with its interactivity. You rely on the interactivity to sort of bring out what your sample has. While it's really good for a single sample, trying to compare this information dense pie between two samples will be really hard on the human eye. Right? So Because of that, me and Dr. Gitanjali have been working over the past 2 months for an alternative visualization. We are calling it the FlumenPlot. Essentially, it's an interactive Sankey plot. So here, how this tries to depict the data is we go from the highest taxa rank to the lowest taxa rank from left to right, where each taxa flows into the other taxa. So you know what the relationships are, and the length of the bar depicts is its relative abundance. So it's really good at showing you both relative abundance and the composition of the sample. And it makes cross-sample comparison a bit more easier, I would say, than Corona, because it's you can control how dense the plot tends to become. But as with any plot, it has its limitation. It's not conventional, so it might take a few tries to get used to it. But if you get used to it, I would say you can get out valuable information much faster than using a chronoplot. But again, it tends to become very dense when you use highly complicated data set, when you have like thousands of species that have very similar relative abundance. It gets a bit cluttered, but that's why we made it interactive, so that you can sort of filter out, make sure the visual fits to your requirements. But yeah, that's been me. Thank you very much. So for the demo session, we will look over what how the interactivity works for the Flumen plot. I have prepared 5 interesting samples of the human gut. There 2 are from Peru, 2 are from Fiji, and one's from Ethiopia. So you guys will have a chance to look at these samples and draw your own inferences. But yeah, we'll start in like 2 minutes. Okay, I guess. So I will quickly give a demo on how to use the tool. So this is also a CLI tool, but we've made a web server out of it, so it's easier to use for non-technical people. So we have 3 tabs. We can click on Get Started. It explains how to use the tool. You require the output in this specific format. Uh, you go to this website, db.nipgr.ac.in. Yeah. Uh, Okay, so you guys prefer to follow along. Okay, all right, sure, sure. Sorry for that. That's db.nipgr.ac.in/metagenome, metagenome. Uh, everyone, okay. Ma'am needs help here. Third row. Okay. Yeah. Oh, cool. I'm guessing that's the last person. Okay, so this should be your homepage. You can head over to tutorial, and then you can click on the example file button to download the example file. It should save as example.mpa.txt. Uh, it should save as example.mpa.txt. On the tutorial tab, the step 1, there's a example file button. Yeah. And then we go over to the FlumenPlot tab. Here we choose the file we just downloaded. The example.mpa. Some of you it might say .txt, some of you it might just say example.mpa. And then you'll see a green tick here to know you've picked. Then you click on submit. Yeah, so did everyone get the plot? Okay, so the MPA file is the default format for a classifier. So when you use a metagenomic classifier, your output is an MPA file, either an mpa.txt file or a Kraken file. But in most cases, you can get anything converted to an MPA file. It just stands for metaflan output, metaflan, which is another classifier tool. Yeah, you got the plot, right? Yeah. So here is just a preview of the plot. So we are packaging the plot as a HTML file, right? So that way, this plot you can download, email it to friend, take pictures of it, save it, preserve it. So you can click on download HTML and it will save the result.html. Yeah, a button should appear. So then we click on result. It opens up as its own thing. So here, is everyone on the result.html? Anyone having any troubles? Yeah. Yeah, it's the same plot. Yeah. That's just a preview of the plot. Right. So if everyone's on the plot, we can see a couple of buttons on the top, right? We have 4 different buttons. We have 5% read, 1% read, and then 0.5% read. So these help you filter the sorry. Yeah. So these stand for relative abundance. So these filter your sample for taxa which are above this threshold. So if you click 5%, it'll only display taxa which are above 5% in relative abundance. So the most dominant taxa. Abundance. It's not better, it's more present. So imagine a pie chart, right? So you can represent that as numbers or you can represent it as a percent. So if the percentage is high, it takes up more of the pie. If it's less, it takes less of the pie. That's all it means. And then we are filtering the taxa here per sample to be above 5%. So if let's say streptomyces is present at the abundance of 6%, it will be visible when you click 5%. But then if it's below it, it won't be visible. So yeah. So first three let you pick the abundance percent. So if you click on any of them, it'll update in real time. So you can see how the number of taxa present disappear when you filter it based on these percent values. So at 5%, you have only maybe 4 classes. And then as you go lower and lower, more number of classes become visible, right? Yeah, because as you can see, at 5%, we are only seeing 4 classes here, right? Any higher and we lose out these also. Because the way this works is we are the relative abundance is relative to total reads. It's not to its previous rank. It's not like we have 39% of Pseudomonata. 8% is not 8% of 39%. It is 8% of the entire sample. So that is why we pick such low values. Yeah, right. And that's one way of filtering. And the other way of filtering is based on taxonomic rank. The default is up to class, but then it goes up to family or even up to species if it's prevalent enough. While the example file does not depict it well enough, our other samples will, right? But then the scaling issue I was talking about when you have too many taxa, that happens when you switch to 5% of reads. See, this gets too cluttered. There's too much information, right? Um, Yeah, so I'm looking at ways. So right now we are still finalizing what the filtration methods will be. I am thinking we could limit the amount of taxa you see at per rank. So like when it's so dense and you have so many taxas, we could limit it to the top 20, right? That is something I'm working towards. It's not implemented yet, sadly. But yeah, so that is the interactivity of the plot. And well, you can move these around. If you hover over them, it'll tell you its first parent and its first set of children's. Bit more visible, right? So when it's so dense, you can hover over it to see how it's if it, how it all relates together. So yeah, and this is a standalone report which does not rely on our server at all. The result.html you guys downloaded, that is fully independent. It lets you present your data anywhere. So do we have any doubts on the interactivity? Yeah. Yeah, very, very good question. So ma'am here is asking about this bit here. The amount of empty space here gets cut off because we set a cutoff at 5%. Doesn't mean it's non-existent. Even though bacteria or Pseudomonas is taking up to 52%, only 39% of it is highlighted here. The rest of this might take space less than 5%. We might have maybe 10 different species that are taking 1%, 2%. That's why it gets cut off. But even though it's cut off, the data is still visible for you. So you know there's more information there, and then you can go digging deeper. So yeah. Any other questions? Okay. So everyone understands the premise of the FlumenPlot, right? So for the DIY part, if we go back to the website and go back to the home page, there is a samples button, right? So if you click on it, you'll be able to download it. And once you download it, you're supposed to extract all. Okay, yeah, yeah, you're at home, and then there's a samples button here. If you click on it, it'll start a download for samples.zip. Yeah, it's a zip file. We're supposed to extract everything from the zip file. Yeah, and it'll create a folder, and inside the folder you'll find 5 more MPF files, right? So like I previously mentioned, these are samples. Yeah, sure. So we go back to the home tab, we click on samples, and then that will start download for samples.zip. Yeah, and then you open that folder, the downloads folder usually.
If anybody is having issues reproducing this on your device, just let us know and we'll get to you and help.
Yeah, it has 5 files in it, 5 MPF files. The same example file we used, that was for soil data. Now these 5 files have data of the human gut microbiome. And these are samples from 3 different cities, one from Ethiopia, 2 from Fiji, and 2 from Peru. You guys can try to open it on my server, run the FlumenPlot, and try to compare these FlumenPlots. And because this is HTML, hold on, I'll show you. So again, you go back to FlumenPlot, you click choose file, and instead of the example, you go into the samples folder, and then you pick a sample. Yeah, only one at one time. No. So Yeah, so you can upload multiple samples, but then yeah, but that's not there on the web app. Is there on the CLI tool. So this is the web app implementation. Yeah, 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 yeah. It is possible, but not for visualization. Yeah, it's usually cleaner to keep your samples separate when you're trying to visualize them. Or you can take consensus where you club all of them and make them one sample and not two different samples. So yeah. So yeah, you will get a file like this. You can download it again. And then because this is HTML, you can do this. You can split it across two parts of your screen and then ask it to redraw it. If you click the buttons again, they should be able to redraw it. So that will let you compare them easily. So try to compare your 5 files, see if you find anything interesting, and let me know. MPA is stand for the format name metaflan output. Metaflan is a classifier. Yeah. So you have different types of classifiers you use, which we started with Kraken 2, which is K-mer based, which looks for your hidden Markov patterns in your sequence and tries to find where it belongs to, because we get partial information, right? We won't always get full matches. So that was the first tool that was made, which is Kraken 2. And then we have Metaflan 2, which looks for marker genes or marker, instead of genes, it looks for marker patterns in the genome. Like if there's 580s in a sequence that is unique to one species, we can say that, this read might belong to this species. So yeah, Metaflan is that type of tool. Yeah. So were you guys able to open all the 5 files, generate results? Mandeep, Mandeep, ma'am needs help. Ma'am needs help. So yeah, so the output table I showed, that's what these files look like. Any more questions? Yeah. No. After sequencing, you run a classifier. What we do in this step, the taxonomic analysis step, this is where we assign taxonomy to a region, which will give us a table like this. So the number here essentially is each one of these. So how many ever reads we're able to assign to genome A gets assigned a value and its entry in the table, along with its entire taxonomy. So not only does it assign at the species level, it also sort of assigns at each previous rank. So even if it does not know which species it belongs to, it can tell that, okay, I can say with fair certainty that this read belongs to a bacteria at the very least. So yeah, that's how classifiers work. Because traditionally for taxonomy, either you do the amplicon sequencing where you focus on a specific region, which is well documented, but is very conservative, like 16S RNA. So that way you're able to make more confident choices in your taxa. But then when you're doing it like this, when you're doing it with different parts of the genome, you tend to be a little less confident and a lot more consensus based. So yeah. It's not a CSV file. It's the same mpa.txt file. Yeah. Uh, this one? Oh, yeah. Uh, it's just data manipulation. You can convert it to a CSV using like, yeah, but it varies from tool to tool. While tab separated is a standard, if you're using some niche classifier, they might use comma separated. So, yeah. Okay, so I'll take one last round of questions and then I would like some feedback from you guys. Okay, so if no more questions, um, what do you guys think of the plot? Do you do you find it visually appealing? Is it eye catchy? Is it interpretable? Does it make sense? You guys had the time to look at 5 samples, right? Were you able to see how different the samples are from each other? Okay, all right, that sounds good. Um, and any feedback? Would you like to see any more information on this? Yes. Yeah. Okay. All right. Keep that in mind. Hmm. Anything else? Yep. Okay. Angiospermic plants for metagenomic. Okay. Um. Um. Okay. You want to just depict the phylogeny. Okay. You just want a Sankey plot for your taxonomy, for your 50 samples, how they are related. Well, would would be, yeah. So how Sankey's work is, we have to give it two values, right? So one tells it the node and its relationship to another node. Another is a value, which tells it how big the bar are. So unless we have arbitrary genomic distance values, it'll be hard. Yeah, it only works when you have a relative abundance or any sort of abundance. Just taxonomy, you won't be able to see these bars. It'll just be, it'll look more like a network than a Sankey plot. Yeah. Hmm. No. Yeah. Yeah, you could. Yes, the Sankey plot itself, you can come up the Sankey plot itself. So FlumenPlot is what I'm calling it because it is interactive and is taxonomy specific, right? But the base plot underneath the Sankey plot is pretty versatile in what you can depict with it. Um, any hierarchical data, anything which has a hierarchy you want to represent, Sankey can do it. Yeah, but then you'll just have to format it better. Flumen can't do it. Flumen is specific for taxonomy for metagenome. Yeah. All right, guys. Thank you so much for being part of the session. I hope you guys had fun. Thank you.